Hi everyone, this is my oxidative phosphorylation model. So to start off, I want to point out the areas of the mitochondria. Here we have the outer mitochondrial membrane, which is the outermost membrane of the mitochondria, hence the name. Next, there is the intermembrane space, which is between the outer and inner mitochondrial membranes. Next, we have the inner mitochondrial membrane, and the inner foldings of this membrane are what make up the cristae of the mitochondria, which increases the surface area to allow for better exchange of the materials between the environments on either side. Lastly, the innermost part of the mitochondria is the matrix where the Krebs cycle takes place and where oxidative phosphorylation begins. Oxidative phosphorylation is composed of two steps, the electron transport chain and chemiosmosis. All of the NADH and FADH2 that have been produced thus far in cellular respiration start in the matrix. These molecules are where most of the energy from glucose is stored, and they act as electron carriers. They are represented by the white ovals shown here. From one glucose molecule, 10 NADH and 2 FADH2 molecules will have been produced. In the example here, I simplified it to just show one NADH and one FADH2 so we can understand the process a bit easier. Then to start off, we're going to talk about the electron transport chain. In the electron transport chain, these molecules shed their H plus ions, represented by the red circles, and the electrons represented by the red balls of Play-Doh. NADH becomes NAD plus and sheds 2 electrons and 1 hydrogen. FADH2 becomes FAD and sheds 2 electrons and 2 hydrogens. So this leaves NAD plus and FAD, represented by the orange ovals, to be recycled. They can then go back into the other phases of cellular respiration to pick up more electrons and H plus ions. One NADH molecule is capable of producing 2.5 ATP, and one FADH2 molecule is capable of producing 1.5 ATP. At the end, you will see we will end up with 4 ATP molecules since we just have one NADH and one FADH2 that we're starting with in this specific example. Both NADH and FADH2 have high potential energy electrons that shed their energy to pump the H plus ions through embedded protein channels via active transport into the intermembrane space. So here we're going to see the NAD plus and FAD leave so they can be recycled. So we are going to have H plus ions floating around in the mitochondrial matrix. And then we're going to start with the NADH. The NADH enters the matrix at an earlier point, so it pumps its H plus ions through 3 protein channels, and FADH is only going to pump its H plus ions through 2 protein channels. So we're going to see that those electrons are going to pump hydrogen ions through the first embedded protein, and then they're going to hop on coenzyme Q, which is going to act like a little shuttle to bring them to the third embedded protein, where 2 more H plus ions are going to be pumped across the membrane. Then they're going to hop on cytochrome C, where they're going to go to the fourth embedded protein, and more hydrogens are going to be pumped. Now you're going to notice that there is an addition of these purple molecules. So these are molecules of O2. So these O2 molecules are going to split. So we're going to have O and O. And then combining with one O, you're going to have 2 electrons and 2 H plus, and then that's going to make a molecule of water. Now going back to FADH2, we're going to see that it's going to not go in this first protein because it enters the mitochondria or the matrix a little bit later. So it's just going to go through this second protein where it's going to then use the Q as a shuttle, the coenzyme Q or enzyme Q. And then it's going to go onto this third protein and then use cytochrome C, and then it's going to keep pumping. And then it's going to combine with the H plus and the oxygen to make another molecule of water. So this is the H2O that's formed is then released by your body normally in the form of sweat, and the H2O is represented by the blue balls of Play-Doh. The higher concentration of H plus ions in the intermembrane space creates a concentration gradient, which plays a role in the diffusion in chemiosmosis. So during chemiosmosis, H plus ions are going to diffuse from high concentration to a low concentration into the mitochondrial matrix via ATP synthase, which is a protein pump, which is represented by the pink construction paper here on the far right. The energy from this diffusion then phosphorylates ADP and a PI to form ATP. So we're going to see this. So we got 1, 2, 3, and then 4 molecules of ATP. So this ATP is then utilized for the function of the cell that keep us alive. So in conclusion, from one NADH and one FADH2, we are going to get 2 molecules of water and 4 molecules of ATP. Thank you for watching.